Okay, so that was the last of the DNA viruses. We're now going to move on to the RNA viruses. So we're going to talk about some generalizations first. Then we're going to look specifically at the positive-stranded RNA viruses, and then we're going to finish it up by talking about the negative-stranded RNA viruses. The RNA viruses are typically single-stranded, but you know there's always an exception to the rule, and reoviruses are that exception. The negative-stranded RNA viruses will carry an RNA-dependent RNA polymerase with them because they can't make it as soon as they get into the cell because they're a template of the message. Okay? A virion-associated polymerase is carried by reovirus, arenavirus, and retrovirus additionally. Most of these viruses are enveloped, as you'll see. The naked ones, as I'm going to point out, are the picornaviruses, which are all spread fecal-oral, calicivirus, of which the major virus there is Norwalk, and reovirus, which includes rotavirus, all fecal-oral, the only ones that are naked. All right? Your segmented viruses are listed here, but for the exam, the only clinically relevant one that you need to focus on are the orthomyxoviruses, which are the flu viruses. This table starts to cover, just like we did for the DNA viruses, some of the virus families that are positive-stranded and the major viruses that are within those families. Tells you a little bit about the structure and whether or not it's enveloped, etc. And so, again, a really kind of good summary table for the family. And where this is really going to be helpful again for you, for example, if you go over here to the picornaviruses, again, you need to know that all these viruses listed under major viruses are in the picornavirus family. They will test that on the USMLE. So we're going to talk today about calicivirus, the picornaviruses, which is one of the bigger families of RNA viruses, flaviviruses, which are also a large family of viruses that are all arboviruses, togaviruses, which are mostly arboviruses, except for we have rubella virus in there, and then the coronaviruses and the retroviruses. So again, use these tables as a good summary table for you. Again, if you need to look up quickly any information regarding those viruses as far as replication, the tables are a good place to do that. What I want to do, though, is start talking about specifically the positive-stranded life cycle so we can get into talking about the specific virus families and viruses that cause disease within this major group. Now, really, the most important thing you can remember about the positive-stranded RNA viruses are that the positive strand of RNA is equivalent to a messenger RNA. If you remember that, you can work backwards from any of this. Remember, everything else is just mostly straightforward molecular biology. Okay? So let's look at the life cycle. We're starting with attachment like we always have to, penetration and uncoating, releasing the nucleic acid. Then if you look where I'm circling, okay, you have a nice stretch of genome or positive-stranded RNA that is a message. What's the first thing that happens? First thing that happens, ribosomes hop on and we start making protein. So this virus doesn't have to take anything with it. It makes everything it needs as soon as it gets into the host cell. Okay? So it's going to make its own polymerases. What are those going to do? They're going to help it make genome. So an RNA-dependent RNA polymerase depends on RNA and makes RNA. So through a negative-stranded template, we are going to get more genomic RNA. So that some of the proteins that we make are clearly going to be structural. Here's the genome. We're going to package them, and they can get released then from the cell. So again, the most important thing you need to know is, again, that positive strand of RNA is the same thing as a message.